Welcome back! In the last video, we talked about the central dogma of molecular biology, where DNA makes RNA and RNA makes proteins, more commonly known as transcription and translation. In this video, we will talk about what can go wrong during this process, specifically regarding mutations. Mutations happen at the transcriptional phase in DNA. Whether it be some sort of disease or an error in DNA replication, there are various reasons why mutation may occur. Mutations can specifically be described as a change in the DNA nucleotide sequence. There are many kinds of mutations that can occur in DNA. Today, we will be starting with the different types of point mutations. Point mutations occur when one nucleotide base gets mutated to become a different nucleotide base, but it doesn't affect any downstream codons. These mutations will only affect the direct codon that is being acted upon. The first point mutation we will go over is silent mutation. Remember when I said in the last video that coding for amino acids is redundant and there are multiple codons that code for the same amino acid? This is where that knowledge would come in. A silent mutation is when a nucleotide base gets mutated to a different nucleotide base, but the overall codon still codes for the same amino acid, meaning that the eventual protein being made would not be affected. Let's take GCU, or alanine, for example. If a mutation were to occur where it switches the third nucleotide to an A, making GCA, the resulting amino acid would still in fact be alanine. If it were still alanine, the mutation wouldn't have any effect whatsoever on the resulting protein, which is why it's called a silent mutation. This one is pretty straightforward. Next on our list for point mutations is missense mutations. Missense mutations are similar to silence mutations in the sense that one nucleotide base gets switched out for another, but the crucial difference here is that the resulting amino acid does change. This could mean a variety of things depending on the amino acid it turns into. A change from a hydrophobic amino acid such as alanine, GCA, to another hydrophobic amino acid such as valine, GUA, may not affect the protein all that much. However, if alanine turns into glutamate, GAA, which is negatively charged, it will likely lead to serious problems for the protein. Lastly, for point mutations, we have the nonsense mutation. A nonsense mutation occurs when the nucleotide base being mutated creates a premature stop codon. These are similar to missense mutations, but are only classified as nonsense when they make a stop codon. If you remember from our previous video, the three stop codons are UAA, UAG, and UGA. You can imagine how detrimental this could be for proteins if half of it doesn't get made due to this mutation. Let's say that there is a cysteine, UGC, in the middle of a codon sequence, and this gets mutated to become a stop codon, UGA. This would mean that any codons down the line from this one would not end up coding for amino acids, resulting in a smaller protein that likely doesn't do what it's supposed to do. Before we move on from point mutations, I do want to mention a quick tip that might be helpful in the future. Let's look at positions 1, 2, and 3 in a codon. Each of these positions has a nucleotide base. Now let's look at the codon wheel. We see that there are quite a few instances where silent mutations could happen, but I want you to notice where these mutations would be occurring in the codon. Silent mutations typically occur at the third position of the codon, while missense mutations are more likely to occur at the first and second positions. You can use this as a mental shortcut when trying to figure out how a mutation would affect a codon. Alright, now we understand that there are mutations that can swap one nucleotide for another. However, that is not all that they can do. They can also insert or delete a nucleotide base, throwing off all of the downstream codons. These are called frameshift mutations. Let's start with the first frameshift mutation, insertions. Insertions, as the name implies, inserts nucleotide bases into a codon sequence. Think for a second about how this would affect the protein. Look at these two codon sequences. Identical, right? Now let's add one nucleotide base anywhere in the bottom sequence, let's say here. We still have to read the sequence by 3 codons at a time, so notice how every single codon downstream of this just changed. Frameshift mutations tend to lead to very significant changes in the protein, usually resulting in it being completely non-functional. A very similar circumstance occurs with deletions, but instead of inserting a codon, you guessed it, one gets deleted from the sequence, once again throwing off all of the rest of the codons downstream. Now, these frameshift mutations can insert or delete more than one nucleotide base at a time, but you will likely not come across this too often in your lectures. Regardless, it's the same process and shouldn't be hard to figure out. If 2 nucleotide bases are added or deleted instead of 1, the protein is still probably not going to be very functional. However, if 3 bases get inserted or deleted, there is a chance the protein will be functional since it just added or deleted one whole codon. This means that there are likely no downstream effects. Now, there are obviously exceptions to this, such as a stop codon getting deleted or introduced, which would be very problematic. But these are very specific problems that I'm sure you can figure out as long as you understand the different mutations and their effects. And that about wraps things up. I know this was a shorter video, but I hope it had enough content to give you a good head start on mutations. Thanks for watching! In the next video, we will discuss the cell cycle, mitosis, and meiosis.